All right. Well, thank you very, very much. That was a lovely introduction. Made me quite nostalgic, actually. And it's a pleasure to be here in Hungary. I've been only once before in Hungary. It was about 20 years ago. And I didn't begin to see all of the beautiful sights there are in the country, and I'm catching up just now. I will go through some of the early education stories that I remember, and you've heard them touched on already. I'll tell you how I got to the core of the work that I did, how I changed in mid-career from being essentially biochemically oriented to being a geneticist, and also about the work we're doing just now. I'll spend some time with that, and I mean for this to be a talk that more or less anyone can understand. As you heard, I had an early start in science. My father was a biomedical scientist who was quite well known. I was interested as a child in nature, and at some point he talked to me and he told me that what I was doing, merely observing nature, wasn't the same as science. One had to ask questions, to do experiments, to try to understand how things really operate. And he invited me into his laboratory when I was 14 years old. I remember in the early days learning quite a bit about red cell enzyme metabolism, doing spectrophotometric assays that were linking two enzymes together in order to measure the activity of one, adding an excess of the other. I learned how to calculate Vmax and Km, and I learned also, because many people in the laboratory were doing that, how to separate proteins, either by isoelectric focusing or column chromatography or size exclusion, and I began to feel confident of my abilities to do that, to purify proteins, which was very important to me only a few years later. It wasn't only in my father's lab that I worked. I had experience in a Drosophila laboratory run by Dan Lindsley at University of California, San Diego. He was an eminent Drosophila geneticist who was interested mostly in spermiogenesis and mapped mutations that caused a failure of spermiogenesis. And I learned from him how to map phenotypic traits to visible phenotypic markers or intervals between such markers on a chromosome. One couldn't go much farther than that in those days, in the mid-1970s. But certainly that was very important to me later on, because I felt when it came to doing the same in mammals, it should be not really so much more difficult. Actually, it was, and we'll come to that in a while. I worked, too, in the lab of Abraham Braudy, who was an endotoxin biologist interested mainly in treating endotoxic shock by passively immunizing people with antibodies against endotoxin, which his laboratory was developing. At the time, you don't really think too much about things that you're learning and whether they'll be valuable to you later on, but I'm sure that it was from him before medical school that I learned endotoxin was something inflammatory that could cause shock and organ injury, something to be wary of. It was a small molecule, it was heat stable, it could only be destroyed by baking glassware at 180 degrees. And I never imagined that I'd really focus on the whole question of how endotoxin worked a few years later. I suggest the same might happen to any of you, that you hear something quite casually, you later become quite dedicated to it in the course of your scientific careers. From Susumu Ono, I learned about mouse genetics and about immunology. He was interested especially in the major histocompatibility complex proteins and what they were there for. It may seem strange nowadays, but nobody understood what they did. Ono had the idea that they were anchorage sites for proteins that direct organogenesis, which is completely wrong. Nonetheless, I learned how to use current methods, complement fixation, and other assays that one could use to quantitate proteins that would bind to the MHC and to measure the MHC themselves by cytotoxicity assays. It came to a question after college what I should do, whether I should go to graduate school, because I was very interested in science, or to medical school. And again, my father gave me some advice. He said, definitely you should go to medical school. There you'll learn about pathology, about physiology, about pharmacology. You'll study one organism intensely, and you'll come to understand what it takes to keep an animal alive, essentially. What are the basic requirements for life? Then you can do anything you want in biomedical science or any aspect of biology. And I asked, what about an MD, PhD degree? And he said, no, that's not necessary. As long as you're not Mr. Beutler, you'll be able to compete for funding and you'll be able to do whatever kind of work you want. And that more or less was a prediction that was fulfilled. He also said, if you don't do well at science, you can fall back on being a doctor. And I always thought that was not very good advice, although it was well meant, because if you do something with an exit strategy, maybe you don't work quite as hard at it. In medical school, of course, I had a lot of intense experiences, and I learned really what it was to be a clinical doctor. I also learned quite a lot of basic science from Bernard Reutzmann and Patricia Spear. I worked in a laboratory there too on herpes viruses and got to know quite a bit about how to work with viruses. But this person comes to mind. You've probably, many of you heard of Kaplan-Meier plots, which introduce the factor of time into variation of two different samples. For example, a treatment group and a placebo group may have different responses and different survival times in a particular disease, and he is the Meyer of the Kaplan-Meier plot. I hadn't thought too much about statistics prior to that time, never had a statistics class in college, which probably I should have. But from Meyer, I learned quite a lot about statistics, and I remember he introduced me to R.A. Fisher's work, and Fisher in particular was quoted here as saying, "Every experiment may be said to exist only in order to give the facts a chance of disproving the null hypothesis." And he emphasized that hypotheses are never proved. They can be disproved. And if you disprove a null hypothesis, then you can be satisfied that you have a certain effect going on. And you'll see that that too was really important to what we do today. It was after medical school that I decided I certainly wanted to go back to science, but I did want to become a finished and a little bit more experienced physician. I went to the University of Texas Southwestern Medical Center to be an intern and a resident, first in internal medicine, then in neurology, because I wanted a really rigorous, challenging experience, and I heard that that was the toughest residency in the United States, and I wouldn't be surprised if that was true. Partly it was good for me because I came away from that with an intern's mentality. At least I expected to have to work very, very hard, and frankly, I could just outwork other people in the laboratory when I went back to the lab. But I did go back to the lab. I really missed working in a laboratory. I went to Rockefeller University to the Cerami lab in New York City, and there the lab had an interest in wasting in chronic disease, which of course I had seen as a physician. And it was felt that it was perhaps cells of immune origin, like the macrophage in particular, that sounded an alarm when an infection was present and made a catabolic factor that would shut off the biosynthesis of fat or the uptake of fat and its storage in fat cells. The relevant enzyme for storage is this one, lipoprotein lipase, which cleaves triglycerides and allows then the fatty acids to cross the membrane of fat cells where they become reesterified and stored. And already the idea had migrated a bit by the time I arrived in the lab. First, it was directed toward looking at factors from trypanosomes that might activate the macrophage. And in mice, that didn't really seem to be fruitful, nor did factors from cancer cells, although there was the idea that that too could trigger cachexia. Instead, when I arrived, I began using endotoxin to activate the macrophages, and I found that this produced an abundant bioactivity that was heat sensitive and that would act on pre-adipocytes that I had differentiated to fat cells to shut off lipoprotein lipase by 90 to 95%. It was a good assay, quite a difficult one, a biochemical assay, but I had this background of purifying proteins, and I understood what I had to do, and I grew up large cultures of mouse macrophages of a permanent line called RAW264.7. I stimulated them with LPS. I would harvest liters of supernatant from these cells, and I would fractionate the supernatant and test for the presence of cachectin bioactivity in 3T3L1 cells. Doing that, in a few months, I purified the cachectin to homogeneity, and this is what it looked like on a gel. It was about a 17.5 kilodalton protein in subunit size, and it seemed to be a multimer when I looked with gel filtration methods. It wasn't long until I was able to sequence the N-terminus of this protein, as we would do in those days, and I found that it was very similar to human tumor necrosis factor, which had been isolated by workers at Genentech, who actually had quite a large division devoted to that, and they found this protein and isolated it by looking at its ability to kill tumor cells. So I did cross assays, and I found that indeed what I had as cachectin was the ortholog of human TNF, but I had isolated it from mouse in response to LPS, and I understood that it might be responsible for many of the effects of LPS. At least that was my orientation. I found that it was very toxic if you gave it to mice intravenously. About 25 microgram would kill a mouse, usually, and on the other hand, a heat-denatured form of the protein would not. Furthermore, I made an antibody against TNF. I passively immunized mice against TNF, as we were then calling it, and I showed that this would shift the dose lethality curve in response to LPS very significantly to the right. And that meant to me that this was one of the major mediators of endotoxicity. It was probably important in many different forms of inflammation. I also used radio-labeled TNF to find receptors for TNF on the surface of many different cells. I felt that practically all cells responded to TNF in one way or another, or could bind it at least. And because these curves were biphasic, I felt that there must be at least 2 receptors for TNF, which proved to be correct. There are exactly 2. With that in mind, I left the lab at Rockefeller, started my own lab at UT Southwestern with the Hughes Institute, and one of my initial goals was to make a synthetic inhibitor for TNF, because I thought that neutralizing it in the course of endotoxic shock or perhaps chronic inflammatory diseases would be useful and clinically beneficial. I felt that antibodies wouldn't be the right tool for this job because I thought, wrongly as it turned out, that antibodies by themselves would be immunogenic and couldn't be used for a long period of time. How could I make a non-antigenic, stable binding reagent for TNF that would neutralize it? Well, the receptors that I had looked at by binding analysis were ultimately cloned by Wallach at the Weizmann Institute and also at Genentech. And once their sequences were available, I thought I will link them to an antibody heavy chain that will force dimerization of the receptor. I knew that this was a trimeric protein, and I even knew its structure at some point because I worked with Steve Sprang in Dallas to crystallize TNF. And this is a mock-up of what I had made in the laboratory when we did that. This would be the receptor binding domain, this would be the antibody heavy chain, and we interposed a thrombin cleavage site that let us analyze what was going on, and indeed we could deduce that this exact sort of structure, binding of the receptor to the TNF, must be taking place. The other possibility would be that our molecule had linked TNF into a big network solid, but it didn't seem to precipitate TNF. It seemed to remain soluble, just it was rendered inactive. Now, this reagent eventually was patented by us and licensed, and it became a very successful drug known as Enbrel or etanercept, and it's been given to millions of people with psoriasis, as you heard, also rheumatoid arthritis and ankylosing spondylitis and some other diseases, and it can make quite a dramatic difference in clinical course. While I was doing that, I had another project in mind, and I began working on it, but quite unsuccessfully at first. I wanted to understand how TNF synthesis was regulated. And of course, I was using LPS as a stimulus, so really I knew all along I was studying LPS signaling, and there was really very little known about that. LPS as a substance had been around for 100 years or so, since it was named endotoxin by Richard Pfeiffer. And every medical student knew that it would provoke shock with bleeding, uncontrollable bleeding intravascularly. The coagulation was upset. There would be injury to many organs, stiffening of the lungs requiring intubation, very serious and quite common problem. It was really quite widely believed that endotoxin must have a receptor, not least because of certain genetic evidence that I'll show you in a moment. And what that receptor was, nobody seemed to be able to find. Many people had looked using protein isolation techniques. And I tried that approach myself and got nowhere. I also tried using expression cDNA cloning and got nowhere. But it was really important to find that, because when you came down to it, the initial events during an infection that lead to this type of inflammatory response were completely unknown. And for that matter, the origins of most inflammation were unknown at the time also. It happened that there were certain strains of mice that were completely refractory to LPS because of spontaneous mutations that by chance became fixed during inbreeding at the major mouse vendors. One of these strains was called C3H-HEJ, and the other was called C57BLAC10-SCCR. In each case, there was a control strain that looked identical to the refractory strain and was genetically very similar, having been isolated only a few months or so removed in time from the observation of a refractory state in the other member of the stock. So we had these tools, and if one could find the mutations, then one would know the gene and one would know the key protein that was required for LPS signaling. Most people thought, and I thought, it was probably the receptor itself, because this was something very specific for LPS. It didn't apply to poly-IC, for example, or to double-stranded DNA, or any of the other inflammatory things that were made by bacteria. By 1978, it had been shown that these two strains of mice had allelic mutations. The same gene was affected by each of these two spontaneous mutational events. And the mutation in the C3H-HEJ strain had been mapped to mouse chromosome 4 by Watson and Riblet in 1978. But they had dropped the problem more or less, and it had just been out in the open in all the intervening time. I was kind of perhaps too self-confident, but even though it was an industrial-sized project, I thought I could track down this mutation and I could find it. The way to do that is through a process called positional cloning. The goal of positional cloning is to show by meiotic recombination that the mutation one is interested in resides between two markers on a chromosome. And these markers were not visible markers anymore, but molecular markers, microsatellite markers. We identified them and narrowed down the interval as much as we could. And finally, we had a marker called B and another called 83.3. And in each case, we showed that there was a crossover between B and our mutation of interest, and between 83.3 and our mutation of interest. In a very large panel of mice, we looked at more than 2,000 meiosis in order to make that determination. This was a large interval of DNA, about 5.8 million base pairs, and we cloned all of it into large chunks of overlapping DNA called a contig, and then we started in the middle of this contig and began fragmenting the DNA, cloning it into sequencing vectors and sequencing it by hand, working our way out toward the edge. Everything in yellow is sequenced completely, or was sequenced completely by the time we finished the project. Everything in pink had been mostly sequenced. And we were sequencing by hand and could look at only a few thousand base pairs per day, and then we would blast the sequences and look for genes. And this was quite tedious, but it was like prospecting, I would say. It was the case that every day you knew you might find the gene in question. But for 3 years, we found only pseudogenes and fragments of a real gene in the contig. And by the end of that time, I was getting extremely nervous because other people were working on the same problem. We had found nothing. We didn't really know what stage they were at in their work. And it was always possible that somebody in the lab had made a mistake in the mapping, and really we weren't even looking in the right region. But I felt that we had to find the mutation very soon because we were running out of DNA to explore. And sure enough, one day, as I was going over the blast results at night, I saw a very strong match between an RNA sequence in a database and a piece of our contig, which suggested that there was a gene in that region that was a better match than any of the pseudogenes. The gene was named only with an alphanumeric, TLR4, toll-like receptor 4, named for its similarity to the fruit fly toll protein. I got excited as I looked at the toll protein because it had leucine-rich repeats, and that made it a good candidate for reasons I won't go into. It also had a cytoplasmic domain that was very similar to that of interleukin-1, and interleukin-1 is an inflammatory cytokine, and it binds to a receptor with that kind of domain that I was seeing here. And it activates NF-kappa B, one of the key inducers of TNF, among other cytokines. We dropped everything and we began sequencing the cDNA, which we amplified the very next day. And in due course, we found a single nucleotide difference that distinguished C3HHEJ from its control strain, C3HHEN. And this is exactly how it appeared to me because I photographed the screen, was very excited that we'd really found the key mutation. But this wouldn't have been enough to publish, I have to say, because others would have argued maybe that's just a passenger mutation that crept in during the last 35 years since people identified this strain. Recall, though, we had a second strain. In that case, we couldn't amplify the cDNA. It wasn't present. And eventually, we defined a genomic interval exactly 74,724 base pairs in length, with all 3 of the exons of the gene residing in that interval normally, and they'd been cut out by a deletion of that length. So that was definitely a deleterious mutation. The other one probably was, and the control strains had neither of these mutations, and we were convinced that we had found what we were after. On the very first night that I saw the TLR4 in the contig, I did blast searches, and I found there were several other toll-like receptors in the mammalian genome, also all alphanumerics, and some of them not even named yet. And these now are known to number 10 in humans, 12 in the mouse, and 13 in both species together. It made sense to postulate that they acted as receptors for different signature molecules that herald infection. LPS is one of those, but there are also bacterial lipopeptides, flagellin, nucleic acids of microbes, while they're chemically quite similar to those of mammals. They have some differences in terms of how much they're methylated at certain residues, and this is enough for these receptors to distinguish self from non-self. I also found that not only mammals, but also fruit flies had a number of paralogs belonging to this family, and I was aware from the very first night, too, that Jules Hofmann, a Drosophila geneticist in Strasbourg, had demonstrated about a year and a half earlier that in fruit flies, mutations in toll, or its protein ligand called Spätzle, would cause the flies to be immune compromised in a particular way, so they became overgrown with fungal hyphae if you inoculated them with that. Now, the mice we were working with were very sensitive to gram-negative bacteria because they didn't recognize endotoxin, and I thought that was a parallel situation, and this was probably conserved in the course of evolution. What I didn't expect, though, was that even plants had similar motif-bearing proteins, cytoplasmic proteins with elements similar to the cytoplasmic domain of the toll-like receptors, and in every case, these proteins were involved in disease resistance in plants, resistance to fungal rusts or bacterial blights, and this was clearly something that was conserved throughout the whole tree of life. I loved this whole approach. I was just enamored of going from a phenotype to finding a critical molecule, and I wanted to do more of it. I moved shortly after that to Scripps Research Institute, where I could set up a program that enabled me to make new phenotypes. How do you do that? Well, you give a germline mutagen to mice. What is that mutagen? It's ethyl nitrosourea, or ENU. You give it to male mice. It mutates their spermatogonia. They pass mutations in the sperm to male offspring, which you can then harem breed to black 6 females, and you get then daughter mice to which you can cross the same G1, and that inbreeding step can bring these mutations to homozygosity in the 3rd generation, so you can detect recessive phenotypes. We knew when we started this that we were having an effect because we saw all of these unusual-looking mice in the 3rd generation, but mainly we were interested in immunologic phenotypes. Still, we tracked down everything that looked abnormal as well because we had the ability to do it, and some of those led to very interesting stories. We solved about 300 phenotypes in the course of 11 years. Keep that number in mind because the situation has changed. Those were exciting times. We saw bizarre animals that no one had ever seen before, in effect, and in every case, the phenotype came down to a single mutational difference, in every case in a coding region, not in intergenic regions, for example, or in cis-acting regulatory regions, and that was important too. This is a mutant called Pinky, which loses all of its fur after it turns gray, gets a kind of dermatitis with inclusion bodies, has bone deformities. It has a problem with the co-receptor for vitamin A. The knockout is lethal. This mutation is viable, but it creates this phenotype that looks quite bizarre and has some immunologic sides to it as well. We found mice with spontaneous inflammation of their feet, and this too was tracked down, in this case, to a phosphatase. Again, the knockout is lethal, but this viable hypomorphic mutation gives you an immune disturbance. There were mice that lost most of their fur and had a severe iron deficiency anemia. Give them large doses of iron, and they would regrow their fur. The females, formerly sterile, regained fertility. This phenotype was similar to something known as iron-refractory iron deficiency anemia in humans, which had not been solved genetically, but we were able to map the phenotype to an interval and find that this was a mutation in a protease called TMPRSS6, and in humans, too, TMPRSS6 turned out to be the causative gene in which mutations create this phenotype. We found new causes of vestibular disease and deafness in the mice. Let me see if I can show you what that looks like. Mice with vestibulopathies run in circles. They tend to be hyperkinetic, and they are also deaf in most cases, as in this case. This, against all predictions, was a mutation in a new catecholamine O-methyltransferase, normally involved in catecholamine metabolism, but in this case, affecting hearing selectively. And again, in humans, there were several families in which homozygosity for mutations in the orthologous gene caused deafness. Then we had a very strange phenotype that I can't resist showing you. We called it possum for reasons that'll be obvious. A technician was working with this mouse, and she picked it up, scruffed the mouse, as we say, just as you see here. This is the proper way that we handle mice. And she put the mouse down and thought she had killed it because it was just motionless. It stopped breathing. The tail remains stuck up like that and has kind of waxy flexibility. And in reality, the mouse is conscious. We did electroencephalography on it and found it has completely normal brain waves, and it's presumably aware of everything that's going on, but it's paralyzed in this way, and you can roll it on its back, and it won't really object. It begins breathing again quite soon after this is done, and minutes go by, and then I'll try to speed this up just a little bit for you. If you come back and you prod the mouse just a bit, as will be done in a moment, the mouse will get up and walk around again. Let's be patient. We'll see that happen. There, you see, it's fine. And you can repeat the process. And according to people in my laboratory, the mice don't seem to like this. They get a little bit pissed off about it, but they're basically unharmed. Now, this is a mutation that we mapped to a voltage-gated sodium channel that's associated with the perception of cold-induced pain. It's a hypermorphic mutation, not a loss of function, and it works as a dominant phenotype. And only scruffing will provoke that phenotype. The gene isn't expressed in the central nervous system at all. It's only expressed in dorsal root ganglia. And why it gives that phenotype, I do not understand. And I wish somebody in neuroscience would work on it a bit more. What we do know also is they develop bradycardia of a severe degree that lasts as long as they're not mobile, but their blood pressure is maintained, and that can't explain the phenotype that we see. As I said, we were mainly interested in innate immunity, and over the course of those 11 years, we found 77 mutations that induced phenotype, tracked them to 36 genes, because we had multiple alleles, as you see in this illustration, and 11 of these genes were new participants in the toll-like receptor signaling pathways. We use TNF as our indicator of response, and sometimes interferon as well. We found hits in all of the receptors that we kept under surveillance, new adapter proteins, certain chaperones that are needed to get the TLRs where they need to be in the cell to signal, and we also found things that would process TNF itself. Now, all this was very exciting for me, and I loved the technique still, but it was much too slow. It had taken us 5 years to positionally clone our first gene. Even with massively parallel sequencing, it could take a couple of years, and there were many false positives and false negatives. The rate-limiting step in the whole thing was the meiotic mapping step. Let me explain how things would be if you were working in my lab in those days. You might be assigned to a screen looking at TNF production in response to LPS, and this is a real example. You would have done about 4,000 assays, and then you'd find an extreme outlier, and you'd ask yourself, is this some kind of mistake, an artifact? I've been doing this thousands of times. I'm only human. How do I know I didn't just make a mistake? To know, you would have to outcross this mouse because you couldn't repeat the assay. You would find the next generation of mice derived from it have normal responses, but that would be characteristic of a recessive phenotype. And then if you backcross to the founder and saw that you had the phenotype again, you'd be confident that this was no error. But you wouldn't be confident that it was something new. It might be just another allele of TLR4, and that would certainly disappoint you, especially after you had gone through all of this trouble by that time. To find what it was, you would have to make a large stock of mutants, outcross to a mapping strain, backcross to the stock, then genotype and phenotype hundreds or perhaps even thousands of mice, and then ultimately you'd be able to say the mutation is in this new gene here. That sometimes took years. Furthermore, you would miss a lot. If you had a situation where you were making large pedigrees, which we should have been doing all along but couldn't afford in the beginning, you might see two outliers in one family, and you would say, whether rightly or wrongly, I wouldn't make a mistake twice in one day. You might then set about immediately to map these two mutations by the same technique. But let's assume everything was all the same, except the effect size of the phenotype was as shown here, rather than an extreme pair of outliers. Let's assume they looked like that. You would look at that pedigree and you would say, there's nothing here for me to find. I will move on to the next pedigree. But you might be wrong. And it occurred to me that if you had a magical kind of tool that would let you say of this pedigree, somewhere in this pedigree, there is a mutation for which these two mice are homozygous, or VAR in our jargon. These shown in green are heterozygous, or het, and these in yellow have homozygosity for the reference allele, or ref. Then you'd look very differently at that pedigree. You'd say, this is something that wouldn't happen by chance, this kind of arrangement of zygosities. And indeed, these days we do have that tool. This is what automated meiotic mapping is all about. And you have the data presented to you for every mutation in a pedigree in this format with columnar separation of the populations of different zygosities. We began doing this in 2013, and at once we began finding mutations hundreds of times faster than ever before. As ever, we use ENU to mutagenize mice, and we make G1 offspring. We take the males because we can breed with many females at one time to generate G2 mice, but these days we make many more G2 mice and even more G3 mice by backcrossing. The key is that we first sequence all of the G1 mice to find what mutations in coding sequence they have up front. And then we genotype all of the offspring of the G1 of both G2 and G3 generations to determine the zygosity of every single mouse. Only at that point, when we know the zygosity of the whole pedigree, do we submit these mice to a phenotyping pipeline. And they're examined with about 100 different assays over the course of about 3 months. We look at weight, at innate immunity, at adaptive immune function, also at growth and development, at blood pressure, heart rate, homeostatic responses to a cytotoxin. And as I'll tell you later, we look for resistance phenotypes, resistance especially to cancer. Transiting this pipeline takes about 12 weeks. 100 assays, as I said, are performed on every mouse to detect phenotypes, and the average pedigree harbors about 60 coding or splicing changes that are non-synonymous. We then can look at about 600 mutations per week in 10 pedigrees fed into this pipeline, and that means that we're doing 60,000 assays of each mutation with regard to phenotype each week, or about 3 million assay mutation pairs each year. And each assay mutation pair tests the null hypothesis for a mutation, which states the zygosity of this mutation has no influence on the outcome of the assay. And usually that is not rejectable, but sometimes it is, as you'll see. We don't only use statistical computation, but we also use artificial intelligence. There are a total of about 67 features we can look at when we assess the data. One of them is a kind of conglomerate feature called the human algorithm. Others have to do with separate artificial intelligence assessments as to whether a mutation damages the protein and whether that protein is essential for life. But we use this diagnostic to tell us whether some kind of statistical excursion from expectation is really likely to be causative of the phenotype. And one always uses a diagnostic, assesses a diagnostic with a receiver-operator characteristic curve. The area under our curve is about 0.95. And this program has been trained on experiments where we take a candidate causative mutation, we recreate it in a fresh background mouse, and we see whether in an expanded pedigree it really is causative. That's how the AI learns. At present, it's much better than a human observer, and the score you get from the artificial intelligence assessment, the CE score, corresponds in terms of this curve to the real probability of verification of a mutation. These days, we only look at things that are at this end of the curve because they're very likely to verify, and we reason if we keep something under mutational pressure, if it's in the iffy zone here, if it's really true, we're going to hit it again and its prospects will improve, and then we'll go after it. What do we actually see when we are mapping mutations? Well, first of all, I get an email every 6 hours that directs me to results that look like this. This is a Manhattan plot. Every dot is a different mutation going across all of the chromosomes of the mouse. There are on average about 60, as I mentioned. This is the negative log of the p-value testing the null hypothesis for each mutation that there is non-involvement with the phenotype of interest. If you were to go and click on this point, the program is interactive, and you'd see the actual data that led to this statistical aberration. And this is the phenotypic performance of homozygotes, heterozygotes, reference allele homozygotes, wild-type mice. You see it again in the lower panel that we have a similar situation. You notice there are a number of other mutations that go along with this one. That's because of linkage on the chromosome. Just to show you that this isn't rare, here are 8 more examples. And usually you have mutations that are linked to others on the chromosome. Sometimes you have one that's all by itself. All of these are highly significant, and for all of them, our CE program says, yes, they're almost certainly real. Here are 50 more examples just to drive the point home. Maybe you're still not convinced that we see these often, so I'll show you 400 more of them. The point is, at this state, we've made 31,359 declarations that this phenotype is caused by this mutation. And that corresponds to 31,359 successful positional cloning projects. Remember, my first one took 5 years, and the whole laboratory worked on it. This is a great improvement. In fact, we calculate that it would have taken us 1,700 years to find that many causative mutations using the old methods that we used during the previous 10 years. At this stage, we've subjected 260,104 coding splicing changes that fall into 21,263 genes to nearly 30 million tests of the null hypothesis. And because we got 31,359 declarations, you can see that about 1 in 1,000 null hypotheses gets rejected and passes the artificial intelligence program that tells us that it's real. Where immunity is concerned, with our very best screens, we've come to about 61% saturation. That is, we've created mutations that either severely damage or destroy the gene, and I won't go into how we know it's severely damaged, but we do. And we've tested them twice or more in the homozygous state. And we can therefore say that since we've identified to date 1,601 genes that are scoring in our best screens for immunity and inflammation, that means that perhaps there are about 2,600 immunity genes in all. Really, there are probably more than that, because we're not testing all of immunity. We're testing a good chunk of it, I think, in our screens, but certainly not everything that one might look for. For dominant screening, we're almost at the same level of saturation. The number above was for recessive screening. Obviously, this leads to the discovery of a lot of new diseases, and some of these we've published, and some of them have been verified in people, And I won't talk about those in particular because at some point we realized that the better use of this technology platform is to turn it around and try to suppress disease. One criticism of my whole approach always has been that most diseases in people, like hypertension, obesity, type 2 diabetes, type 1 diabetes, are polygenic. And this is well known, that they run in families, and yet you don't come down to a single Mendelian cause, nor even a cluster of Mendelian causes. And how does that work? And how are we addressing that? Well, it actually is possible to start with a susceptible background and look for things that accelerate disease or prevent disease. And we decided to do this with type 1 diabetes. It happens there's a kind of mouse that gets autoimmune type 1 diabetes. This is called the NOD mouse, or non-obese diabetic mouse. And together with Lucien Chateauneud and Jean-Francois Bach, we looked at a total of 14 pedigrees involving some thousands of diabetic mice that had been mutagenized on that background. In this case, we don't have a scatter plot, but a Kaplan-Meier plot. And you can see that we found an example here of a mutation that accelerates the development of type 1 diabetes. This is time to the development of glycosuria. These mice get it precociously. And this is essentially a recessive mutation. This would be heterozygotes, reference allele homozygotes in blue. We also found mutations that would completely prevent type 1 diabetes. And in this case, I'll show you what the mutations are. This one is an aminopeptidase. Maybe it's involved in thymic negative selection of TCRs because antigens have to be trimmed before they're presented to T cells. That's just an idea. We don't really know. This one is a mutation in coronin 1A, and that gene is well known to be required for T and B cell development, and these mice just can't develop an autoimmune response. But the point is we have many of them. Some are informative, some are merely confirmatory, and where you find things that block type 1 diabetes, you begin to think maybe we could develop a therapeutic based on that. What about something more demanding? What about giving a lethal cancer to mice, a transplantable tumor? Can we find host germline mutations that make the host inhospitable to the cancer and permit its rejection? Just a single nucleotide change, would it do that? So far, we've looked at nearly 60,000 non-synonymous changes in the coding region. We've surveyed about 10% of all genes, damaging or destroying them, and looking 7 or more times at homozygosity, because this is a little bit of a more dispersed kind of screen in terms of the data that result. And we found 11 cases where we clearly and confirmably have resistance to cancer. In this screen, if a mouse doesn't show any tumor, we don't want there to have been a mistake in the mouse house, and we usually go back and re-inject a second time to confirm that that's the case. Now, one such mutation is called icebreaker. We found resistance in our screen. We expanded the pedigree using the survivors, and we looked with and without checkpoint inhibition and found mice with checkpoint inhibitor that were homozygous for the mutation were very resistant to the melanoma. Even without checkpoint inhibition, they were quite resistant. Heterozygotes and wild-type mice, whether you give them checkpoint inhibitor or not, they all go on to die. This mutation also created resistance to the Lewis lung carcinoma, another very virulent tumor, and 100% of mice that were homozygotes challenged with MC38 would cure themselves. They would reject the tumor outright, and after doing so, they were totally resistant to the other tumors as well. I won't dance around all the data. I'll just tell you how this works. This is a single amino acid change in a certain essential protein, and it changes a presented peptide that's 9 amino acids in length by 1 amino acid. That peptide normally is presented by class 1 major histocompatibility antigen in the thymus, and it enforces negative selection so that T cell receptors don't develop against the wild-type peptide. But in a homozygous mutant, T cell receptors can develop that react with the wild-type peptide in the same MHC context. When a wild-type tumor is given to the mouse, tumors tend to express a lot of this peptide, and that one difference is enough to lead to their rejection. One can follow the T cells that do the rejecting using a technique called MHC tetramer staining. You take MHCs, you make them tetramers, they have the correct peptide loaded into them, and you use that in flow cytometry, and you see in this hexagon, these are the T cells in a tumor-experienced mouse that reject the tumor. Those are in blood, but they're circulating at the level of about 2% of all CD8 T cells. And this is a naive mouse, the same mouse actually before it was given a tumor. You amplify selectively those cells, and they reject the tumor. If you give a tumor again and again, you get to have about 20% of all of the circulating T cells reactive with that peptide, and the mice are simply hyperimmune to any tumor you would give them. Of course, this is reaction with a self-peptide, and I thought if we give these cells to a wild-type mouse, it's going to get some kind of terrible autoimmune disease. We tried that. In fact, we transferred them, tetramer-positive cells, to mice that lacked T cells of their own, and they grew to large numbers in the lymphoid tissues of these mice. And the mice continued to gain weight and look completely healthy, weren't particularly disturbed, did not have autoimmunity. Rather remarkable, but more remarkable was the fact that they were quite tumor-resistant. Not as much as an icebreaker mouse, because there is distracting peptide in the normal tissues of these mice, but very significantly, they would resist the growth of tumors. That was interesting enough to make us want to clone some of these T cell receptors. So we did that. We actually found many of them by sequencing tetramer-positive single T cells, and one of them called TIL3 seemed to be particularly high affinity, and we decided to work with that one. We took the alpha and beta chains of this receptor, we used a retrovirus, and we transfected CD34 hematopoietic stem cells from icebreaker homozygous RAG2 deficient mice, and we then transplanted these back into another RAG2 deficient icebreaker homozygote, and in that mouse, all of the T cells will express just this receptor. And as you might imagine, such mice will reject tumors themselves, and as they do so, they get to have more of the T cells in their blood, and they can serve as a source of these single specificity T cells that can be transferred to other mice. Transfer just 50,000 of those T cells into a mouse with a rapidly growing tumor, if it's an icebreaker RAG-deficient mouse with no other T cells, and the tumor will keep growing for a little while, but after about a week it reaches its maximum size, and then it's rejected just by that one kind of T cell bearing that receptor. On the other hand, if you give normal CD8 T cells to the mouse, the tumor will grow and take over the animal, and if you do nothing, similarly, the mouse will die of the tumor. If you give even a smaller number in recent experiments of these CD8 T cells with the TIL3 receptor to a wild-type mouse, the animal will maintain elite control of the tumor, as it will if you use, for example, just tetramer-positive cells of many different specificities, nearly to the same degree. Now, can this be done in humans? There are complications in doing it in humans, but in principle, yes, you could, to be very quick about it. And we are, at this point, cloning T cell receptors with the same specificity, this peptide being conserved completely between humans and mice. And in fact, the protein is nearly identical and will have very similar biology. We'll see how that happens. Of course, the big problem with humans is they have many different kinds of MHC, but there are some that are very common that do present this peptide. There were other causes of resistance. Here is a mutant called citation, which affects a cell surface protein that's expressed especially by dendritic cells. And at day 13 and at day 20 after inoculation, you see they tightly control the melanoma that's given to them. The mode of action here is very, very different. The mechanism seems to be that without this protein, dendritic cells are much more aggressive at priming a T cell response. These are the cells that activate the T cell. They're innate immune cells. We wondered if that would be only a chronic effect, perhaps developmental, or something acute. So we fluxed this gene, enabling us to knock it out with a drug called tamoxifen, and we did experiments where we knocked out the gene at the time that we gave the tumor, and within that period of time, out to 20 days, knocking out the target gene in all cells of the mouse would lead to this kind of resistance. We also found a perfect phenocopy if we knocked it out only in dendritic cells. So it works in a very different way, and it raised the question, what if we make an antibody against this protein? We did that, and we made sure it would neutralize the protein, and I won't go into detail there, but you see this dose-dependent effect of the antibody on tumor growth. It's not acting on the tumor, which doesn't have the antigen, it's acting on the host and blocking its reticence to make an immune response. Here we have one mutation then that makes a T cell receptor that doesn't normally exist. Another mutation causes aggressive priming of T cells. These two ought to synergize with each other. And to see synergy, you have to use a different kind of cancer model because both give such elite control by themselves. But if you give intravenous melanoma to the mice, that's something that an icebreaker mouse cannot handle, nor a citation mouse. And here you see the black curve is a wild-type mouse. These mice die within about 30 days, mostly of pulmonary metastases to the tumor. The same is true without anti-PD-1 or with anti-PD-1. A checkpoint inhibitor makes no difference. Icebreaker does nothing to prolong life without anti-PD-1. Citation maybe does a little bit. The two together actually are significantly more effective than either alone if you have double homozygotes. If you add in anti-PD-1, then that makes a sensational difference, and the mice live quite a bit longer. And there are still other mutations called whalebone, for example, that also augment the effect of icebreaker and even give survivors in this screen. Of course, there is a little bit of a problem here. We're transplanting a wild-type tumor into a mutant mouse. We're looking only at host-related effects. We're missing any tumor cell intrinsic effects of mutations. You can screen that way, and we have done so, and I'll show you just very quickly one last phenotype that we call glue. These mice have defective immune responses, but not severely so. They have also a decreased number of T cells, NK cells, and NKT cells, and they have also a macrocytic anemia, where they have too little total hemoglobin in their blood, but the red cells are very much larger than they normally are. We thought, for various reasons, these mice might be resistant to models of leukemia, genetic models. So we bred mice that had a transgene for BCL2, which creates a good model of a chronic leukemia where cells just don't undergo apoptosis as they normally should. And we found that homozygotes for the reference allele with a BCL2 transgene develop leukocytosis, but the glue homozygotes simply do not. They absolutely don't get leukemia. We then tried an aggressive acute leukemia model with EMU-MYC. In this case, the IgM heavy chain promoter is driving overexpression of MYC. And these mice usually die within 6 to 8 weeks after they are born of a fulminant leukemia. So we're looking at 4 weeks here. Again, you see that we have lots of leukocytes in the mice with the transgene, but this is canceled completely by glue. And if you look at the blood of a couple of different mice. Here, wild-type mice are showing lots of blasts in their peripheral blood. Glue mice just don't. They're running around the cage. They're perfectly happy. They're breeding. So we regard this as an excellent target. We know this to be a high-level regulator of the cell cycle that no one has noticed before. We know that, in fact, there's not a single publication about this particular protein, but it seems to us a good target for developing a drug, and we're working in that direction now. So, total of 11 mutations in the earlier screen. Glue and also another target revealed by mutations called midnight and full moon affect the proliferation of leukemias in a cell-intrinsic manner. Other things we could do with this platform, we could look at metabolic disease, developmental disease, neurodegenerative disease. I think the most interesting thing might be to look at longevity in total in mice. It would be a difficult screen to do because it would be even longer than the type 1 diabetes screen in duration. But it's already known that there are certain viable mutations that will extend the life of mice by 30 or 40%. And it's my feeling that aging as a general process is something that's programmed into us. We have mutations, we have genes, let's say, that are designed to restrict our longevity, or designed is a bad word, that function to restrict our longevity. And that is why mice live only 2 years or so, and humans can live 80 years or so. If we could do a full genome-wide screen, who knows what we would find and how actionable it might be. Now, when I began my work, my serious work, I had quite a small lab. I would never have gotten the cloning of the LPS locus accomplished without the dedication of Alexander Poltorak, who was very single-minded, just as I was, and wanted to find that mutation and couldn't let go of it. I now have a very much larger group. There are people who make mutations in mice and breed them to homozygosity. There are people then who do the phenotypic screening of the mice, others who sequence the genomic DNA and do the genotyping to assess zygosity of all these many thousands of mice per year, a strong computational group and a strong CRISPR group in order to train the AI. And they all work together, and we are discovering things at a remarkable rate now so that we can really only spend time looking at the very most interesting of the mutations that come our way. I think with that, I'll thank you very much. I'm sure I went over time, but I'm happy to answer questions if you have them. Thanks.
So thank you very much for this wonderful talk, and now the stage is for the students and investigators. Please, questions.
Sure.
I'm not a student anymore, but it was very nice. I was a little bit still shocked about the amount of information. I was wondering whether, as I understand, practically all your screens can only identify functional proteins, if that's, I don't know how, was the correct term, if that alone is causing disease. What happens if, let's say, there's a family of several, let's say, Sark family kinases, or many other families when there's a lot of overlap between the family members? Can you identify those mutations or those?
If there's really true perfect functional redundancy, we're likely to miss a particular gene. It is hiding in the shadow of another gene that does exactly the same thing. We're going to miss that. But that perhaps is rather rare. Only with a very recently duplicated gene will there be perfect redundancy. And usually if a gene has survived, even if it's quite similar, if it survived for a few million years without being crushed by evolutionary pressure, then it does something a bit different. And sometimes, even if there is identity, I suppose having a diminished dose can lead us to find it. We know of one such example where the coding regions of two genes that have recently duplicated are different, and yet we picked up a kind of hypermorphic phenotype from a mutation in that gene. So this is a problem with all genetic screens, but it's not really a serious enough problem to make us stop.
Thank you.
So questions?
Yes, I see you there. It's a case where you fail to have negative selection of T-cell receptors that are specific for a wild-type peptide. And those receptors don't exist in a wild-type mouse because they get negatively selected. But in this mutant, because it differs by one amino acid, the peptide that is different will not cause negative selection of TCRs that react with the wild-type peptide. Is that clear?
Not entirely.
It wasn't obvious for me because I'm an innate immunity person and I had to really think for a while. But you think about that, how negative selection occurs, and you will understand that if you have one amino acid change, that can create a situation where you can get T-cell receptors that have normally been eliminated by the mouse completely. And this is rather rare that the reaction is strong enough to reject a tumor because we've only seen 3 instances like Icebreaker, Icebreaker itself and 2 others in 60,000 mutations we've looked at.
So these receptors have higher variability then, and so they can detect tumors more effectively?
Tumors tend to express a lot of the peptide. We were lucky in that case. And it's enough to cause rejection of the tumor, that single antigenic difference.
Okay, thank you. And so are there any cases where these tumor cells do not express these peptides, and so they remain undetected?
We've done that just to be absolutely sure of the mechanism. We took melanoma cells, we knocked out the gene in question. While the knockout is lethal at the level of the whole mouse, tumor cells can survive it, and they maintain their virulence in the case of B16F10, but Icebreaker is not resistant to those tumors. They grow just fine in an Icebreaker mouse. So it's clear that that's the target. There's no question about it.
Okay, thank you.
Thank you very much. It was really a fantastic talk. It inspired me and had lots of new ideas on my research. And I have one question. You extensively use artificial intelligence to predict if the mutations you find is really valid, gives a certain phenotype. And do you use also the microRNA and non-coding RNA data in your AI system to train, or you just
We don't include those in our genotyping panel. It's come up in discussion a lot. Shouldn't we include microRNAs and long non-coding RNAs and see what phenotypes emanate from them? But that would cost about twice as much to genotype, and we just haven't done it. In fact, there might be cis-acting regulatory elements, though very rare, that create phenotypes when you mutate them with ENU. Why rare? Well, there just tends to be some redundancy of elements that drive the expression of a gene, and it's a common experience that you rarely see phenotype come from those. But if we had all the funding we wanted, we'd just do whole genome sequencing and keep thousands of mutations rather than 60 under surveillance.
So you haven't found any mutations on non-coding regions?
No, we haven't. Actually, they would be quite rare too, because I think such genes are, they're rather tolerant of mutations. There's much less constraint than in coding genes.
Thank you. Questions?
First of all, I would like to thank your amazing lecture, and I'm curious about that the lethal dose of LPS is not body weight dependent, because at the beginning you mentioned micrograms per mouse, but as I know, a wild-type mouse can be also 20, 30, or 40 grams.
I didn't quite understand. I'm very sorry. Can you say it again?
Yeah.
Maybe. Hold the microphone a little farther away.
Okay.
Yes, better.
Is it better? Thanks. So I would like to ask that the lethal LPS dose is not body weight dependent, because you mentioned micrograms per mouse, but as I know, a wild-type mouse can be also 20, 30, or 40 grams.
Okay, got it. What I said was micrograms per mouse was TNF dose.
Okay.
A lethal dose for black 6 mice of LPS is somewhere between 1 and 200 micrograms, something like that. And they weigh about 20 grams, so you can calculate how many milligrams per kilo it is. Mice also are much more tough and resistant to LPS than people are. But in both species, TLR4 is the principal avenue for sensing.
Thank you.
You mentioned that glue can be a new cell cycle regulator. So can you tell me something about its function? So this is normally in the normal mitotic cycle, and you mentioned that maybe it is linked to proteasome degradation. It is maybe targeting some targets. So what is the role of this glue in the normal cell cycle?
Okay, glue, I'll tell you a little more about glue, but don't ask me too many more questions, because my postdoc will get very angry at me. But it's a zinc finger protein. It binds to another zinc finger protein, and the two of them form quite a tight multimer with actually the other zinc finger protein that I mentioned we call plywood. It's a dimer, a tight dimer, and we've solved its structure. And at least 3 dimeric other proteins bind to a single subunit of glue. And then they enter the nucleus where they function. We know that if you have a glue mutation, a viable glue mutation, you get accumulation of T cells in G1, G0, and you have a real deficit of those that are in S phase. And you also have more cells that are dying. So we localize it to the transition from G1, G0 to S. And we know that it binds to a certain motif that is quite common in cell cycle proteins. We know that there's underexpression of MYC, for one thing, but also many other cell cycle proteins, and I think of it as a master regulator of most of those proteins. The reason for the macrocytic anemia is that one of the proteins is dihydrofolate reductase, and these mice behave like they have a folate deficiency. So it's as though you're treating them with multiple chemotherapeutic modalities, but really you've only hit one gene.
Thank you, I think, over there.
Thanks a lot for your great talk. Once you identify potential protective variants against human disorders, do you intend to screen the general human population for similar variants, just GNOMAD database? And if there are you going to compare affected and unaffected human populations for a difference in these variants, if there are similar variants?
That's not the strategy we have in mind. If you think about it, there might be a human icebreaker individual out there somewhere, maybe even a few of them out of the 8 billion people. But if they got cancer, they would not behave differently than anyone else. because remember, they're rejecting wild-type tumors. The therapeutic modality is to isolate human T-cell receptors that react with that peptide in the context of very common MHCs. And that's what we are doing, and it doesn't involve humans at all. It involves humanized mice and human cord blood stem cells to make human T cells in those humanized mice, which also have an icebreaker mutation. That's the way we're going with that.
Thank you.
Thank you. More questions? Students. You pass it.
Thank you. So, if I know correctly, there had been human cases when malignant cancer had disappeared completely without chemotherapy. And is it possible, because it was like from a similar mutation like in the mice?
Yes, yes, it might be. It would not be from something like icebreaker, but maybe from something like citation or whalebone. Or we've observed certain mice with autoimmune diseases created by single mutations that really do have some problems going on, but they sometimes completely reject transplantable tumors. And
can it be mapped like with mice? So can we find that mutation in human genes?
In those later cases, the same mutations might have these effects, these rare effects of spontaneous remission of cancers based on immune mechanisms. And we might find them in humans, but frankly, it's much easier to find them in mice. And I think maybe all of you know why that would be, but just to really drive the point home, we have such success because we're working with mice that are all genetically identical to one another, except for those mutations, those rather few mutations that we put into them. And we know what those mutations are, and we make a large family. This is very different than genome-wide association studies in people, for example. It's real forward genetics, and it's meiotic mapping. It's not purely associative.
So I would like to have another question based on the previous one. So do I remember correctly that it was the icebreaker in the case of which you expected autoimmune disease, but it didn't happen?
Yes, if we took icebreaker cells and put them into a wild-type mouse, just as they reject a wild-type tumor, I thought they would have bad effects on the normal tissues of wild-type mice, but that did not happen.
And what's an explanation for that?
Sometimes a protein just is not very much expressed in the adult. It may be very important for embryogenesis, and yet in the adult animal it is expressed only at very low copy number and may largely escape detection. Also, you have things like T regulatory cells that apparently evolved to detect just isolated autoimmune specificities and dampen them, and that may be the reason.
Thank you very much.
Questions? We still have time for questions.
Thank you. So my question would be if there was a time when you wanted to give up, and if yes, why didn't you do so?
When I wanted to be what?
Give up.
Wanted to give up. Oh. In the case of the cloning of the LPS locus, I never really wanted to give up. I was nervous that we might be beaten in a race to find the gene of interest, but that only made me want to work harder. I think psychologists talk about sunken costs in an investment. If you invest in a certain stock, you may feel that you just cannot sell, or you even wish to invest more and more, having sunk costs, and you wait for the payoff. Or when you are prospecting for gold and you don't find it, and you don't find it, and you don't find it, it just makes you want to continue because you can't bear having spent years of your life and not succeeded. So I guess that's the psychology that made me want to find it all the more.
Thank you.
Thank you. Questions? Still we have time for a couple of.
Well, if I understand it right, you wrote a program for your research, and I got a question that what kind of programming language would you recommend to learn if you want to work in science?
All right, let me be clear. When I was looking for the LPS gene, I did teach myself to program, and I learned the language Perl, because I had so many files to move around and perform operations on. It kept me busy all day. So I made it blast against a database by itself and report significant results to me. And I was never a very good programmer. And therefore, I hired 3 very good programmers. And they had variable knowledge of biology. The first one I hired was really an outstanding programmer, but he knew nothing about DNA, not that A pairs with T or C with G, or that there are 2 strands, but he was a very smart person and he learned all that. And he and most of the other people in my lab nowadays use Python, I believe.
Thank you very much.
Thank you. And one last question. Anybody?
Do you think that one to achieve great successes like you did needs to be born a genius, or is it just hard work and sweat?
No, I don't think I'm really unusual in any way. I'm sure there are people here who are smarter than I am. I think persistence is important, and self-confidence is important. And those are the 2 things I would credit myself with.
Thank you.
Thank you very much indeed. It was a really excellent talk, and plenty of new questions arose. And thank you for being with us. And please let us deliver you the diploma, the certificate by the National Academy of Sciences. So I would like to ask András Varró, the chairman of the curatorial board, to provide a diploma. Thank you very much.